Hi, everybody. My name is Marnie Zerbel. I'm the Research Facilities Coordinator here at Hatfield. We are going to get started in just a minute. Mauricio is going to introduce our speaker, but first I have just a couple quick announcements. For those of you online, if you have any issues, technical issues, please put them in the chat, and our tech support, Katie, will help us help you. And if you have any questions for our speaker, we're going to read those after the talk. And if you're here in the room, please make sure you signed in. And what else? We are recording this event and it'll be online in a couple days. Next week's seminar will be on July 18th when Will White, who's an associate professor with Coombs here at OSU, will talk about climate resilience and the adaptive management of marine protected areas. And with that, I'll get out of the way, and Mauricio, you can take it away.
Thank you. Hello, everyone. Can you hear me? This is often a problem in my life. Okay. Yes. So it's my greatest pleasure to introduce you, Dr. Taylor Hersh, postdoc in my lab and also a long-term friend. Taylor is a bioacoustician with a keen interest in the interplay between vocal complexity and social complexity. She joined us here at Hatfield and Marine Mammal Institute recently in November last year as a postdoc commenter by Professor Kate Stafford and myself, thanks to the Gray Whale License Plate funding. Taylor graduated from the Carnegie Mellon University with a bachelor's in biology and psychology in 2014. And in 2021, she earned a PhD in biology from Dalhousie University in Nova Scotia. And of note, the day she started her PhD back in 2016 was the same day that I was defending mine in the same lab, same university. And although we never actually overlapped at Dalhousie, we did later on at sea doing research on offshore sperm whales around Galapagos on houseboats. So that was really nice. And together we see more water than whales. And we spent a lot, that means we had a lot of free time in an enclosed environment, the place that brings us the best out of us. So two things I learned: that first, cabin fever is real. And second, remember, be nice to people because no matter what, because they may come back and work with you eventually. And then after her PhD, Taylor spent 2 years as a postdoc in the Max Planck Institute for Psycholinguistics in the Netherlands. And there she expanded her research on animal communication by studying rhythm in various species, not only sperm whales, but also harbor seals, African penguins, Java sparrows, and surprisingly, dogs. So in today's seminar, Taylor will share her journey into the acoustic communication in whales and dolphins, some of which has been developed here at Hatfield in our labs. So yeah, let's have it. Without further ado, let's give a warm welcome to Dr. Hersh. I'll keep this.
Okay, how is that for volume? Good? People in the back, good. Okay. Right, so thank you, Mauricio, for the introduction. Thanks to everyone who's watching today. I have sort of a habit in my career of moving places sight unseen and hoping for the best. And I think that's never paid off so much as it has coming here. It's been a really warm welcome from the Marine Mammal Institute and Hatfield, so thank you for that. And so, as you now know, I'm currently a postdoc in 2 research groups. So I work in the lab for animal behavioral interaction research in the ocean, also known as Labyrinto, which is Mauricio's group. And I'm a postdoc in the Marine Mammal Bioacoustics and Ecology Lab, which is led by Kate Stafford. And so I think this is a lot of fun and a lot of work. But what you might not know, although Mauricio mentioned this, is that I've actually known both Mauricio and Kate for about a decade now. So I met them both in 2016. Kate was at University of Washington at the time, and I'd applied to do a master's with her and another professor. And we found out that funding came through for that position 3 days after I accepted a position somewhere else. And so I actually found out that I got a position at Dalhousie, where I had 1 day of overlap with a very bright, talented young man who had just defended his PhD. If we had been there at the same time, this would have been my view because my desk is looking at where Mauricio was sitting. But he had studied sperm whales, and my thesis was essentially picking up where his left off. So I had this goal of quantifying spatial and temporal variation in sperm whale social vocalizations. Now, since finishing my PhD, I've definitely expanded the diversity of species I studied, as Mauricio mentioned, so moving beyond sperm whales. But I think the guiding light of my research remains this goal of quantifying spatial and temporal variation in social vocalizations, and because doing so allows us to learn about the identities, interactions, and cultures of the animals that we study. And it also tells us about the type of information that might be encoded in those different types of vocalizations. Now, this is an ambitious goal because animal communication is really complicated. As some other bioacousticians said, the characterization and abstraction of vocal communication signals remains both an art and a science. And I think this paradox is really nicely exemplified if we look at something like humpback whale song. This is arguably one of the best studied animal vocal displays in the world. We've sort of thrown our best science at it. We know so much about the structure, the composition, the animals that are doing it, how other animals in the population respond, how these vocal displays evolve over time. But there's still room for art and interpretation in our understanding. So this is a somewhat recent, I guess not anymore, a decade-old reinterpretation of humpback whale song as sort of a musical score and trying to use graphics to see if we could pick up on new trends in song that we might not be seeing with all the science that we're throwing at it. So with that in mind, just to give you an overview for the seminar today, I'm going to start by discussing what social vocalizations are and the features over which they can have complexity. I'm then going to talk about the structure of the vocalizations themselves and how we can represent that structure. And then I'll talk about how we can go from these representations of vocalizations to actually quantifying the diversity of different vocalization types. And I'm going to do this by going through a few case studies of species that I've studied at OSU and before, which include sperm whales, bowhead whales, and bottlenose dolphins. Yeah, I knew that was going to happen. Felt wrong. Okay, so starting with the complexity of social vocalizations. What do I mean by a social vocalization? These are the acoustic signals that animals use when they're interacting with each other. And there's lots of types of information that can be contained within those social vocalizations. So that includes things like an animal's emotional state, some indicators of their fitness or their health, and identity. And that could be something as broad as species identity or population identity, or something as specific as individual identity. And those different types of information can be encoded in several different facets of vocalizations or features. So things like the vocalization complexity, the vocalization structure, or the diversity of vocalization types being used. So just to introduce you again to the 3 species that we're going to focus on today, sperm whales, bowheads, and bottlenose dolphins, I just want to quickly introduce the specific social vocalizations I'm going to be talking about. So in social situations, sperm whales produce stereotyped patterns of clicks called codas. Here you're looking at a spectrogram showing 5 different codas. On the x-axis, we have time, and the y-axis shows frequency. And codas can vary in the number, rhythm, and tempo of the clicks that make them up. And the codas in this spectrogram sound like this. For bowhead whales, the social vocalization that I've been studying is song. This is a hierarchically structured vocalization thought to be produced by males in a mating context. This spectrogram is showing 1 song, which lasts about 1.5 minutes. I'm just going to play this segment of it. And then last but not least, when bottlenose dolphins are socializing, they produce frequency modulated vocalizations called whistles. This spectrogram shows a sequence of 4 whistles. 2 at the end are overlapped with each other, and it sounds like this. There's also echolocation clicks in the background. So the 2 axes of a spectrogram represent the domains over which vocalizations can have complexity. So you have time, or the temporal domain, on the X, and frequency, or the spectral domain, on the Y. And so almost all of the research I did during my PhD and in my first postdoc has focused on the temporal domain of vocalizations. I specifically studied rhythm in animal communication, so defined here as a pattern of time intervals that demarcate a sequence of stimulus events. And of course, if we're talking about vocalizations, the events are calls or something like that. And we can have both simple rhythms, which we also call isochronous rhythms, and we can have more complex rhythms, which we call heterochronous rhythms. For hierarchical vocal displays like bowhead whale song, you can have nested rhythms as well. So rhythm at each level of this hierarchy, which gives rise to very complex rhythms. And I'm not going to get too much into rhythm today, but if you find this at all interesting, there is a ton of information that can be encoded in the rhythmic structure of vocalizations. And some colleagues and I have a recent paper out on this if you want to learn more about that. If we then move to the spectral domain of vocalizations, this is a newer thing for me to have to care about. Sperm whales are very much temporal, but what you're looking at here is a snippet of a bowhead song. Kate, I know I didn't put the axes. I know that's a big no-no, but I didn't want to, so I didn't. But what you can see here is that it's a pretty simple note that's being repeated. Hopefully you can get the main takeaway, which is that in this second song on the bottom, you have a lot more spectral complexity in the units that are being repeated. So now we have all of these sort of domains over which we can have complexity, but to actually quantify things, we need some way of going from the real world vocalization that we've recorded to some representation of that vocalization. And to do that, we need to choose features that represent the vocalization. At its core, this is a dimensionality reduction problem. So the goal of which is to compress high dimension data into fewer dimensions while somehow still maintaining the structure and variance in the original data. And if that sounds like a lofty goal, I think it is. It's kind of a like have your cake and eat it too problem. But there are ways to go about this. And what you're essentially doing is you're taking each of your original data points, for us, vocalizations, and you need to project them into a lower dimension latent feature space. So there are 2 ways, kind of 2 main approaches. You can create latent spaces based on predefined features, or you can determine those latent space features by the data itself. And in that case, there are both linear and nonlinear ways to go about dimensionality reduction. By far, the most common approach is dimensionality reduction via predefined acoustic features. And so this is where you transform some acoustic signal into a vector of features. What do I mean by that? This is exactly what we do for sperm whale codas. So here's a spectrogram of 1 coda. It has 8 clicks, and we can calculate the interclick interval for each of those, or what we call ICIs. And then we can represent a coda as a sequence of ICIs. So this is a valid approach if your system is really understood. So for sperm whales, yes. But it does have some limitations. So it requires expert knowledge, and it's prone to human biases because we're choosing what features of the vocalization are actually important. An attractive alternative is to project vocalizations into a low dimension feature space that's somehow determined from the structure of the data itself. Some linear techniques that you might be familiar with are principal components analysis or multidimensional scaling, but there are nonlinear techniques. The one that I'm going to focus on today is abbreviated as UMAP, which stands for Uniform Manifold Approximation and Projection, but we're going to call it UMAP for obvious reasons. And the way this works is that you take spectrograms. So here you see the 3 rows of vocalizations. Those are just some spectrograms of different vocalization types. Treat them as points in some high dimensional space. Build a graph of the relationship between those points in high dimensional space. Embed the points in the lower dimensional space. Do this many, many times. until you find an embedding that best preserves the structure of the original graph. And so those details aren't as important as the benefits of this approach are. The thing that blows my mind is that it can work directly from spectrograms, so you don't have to sort of convert a vocalization into some other features. You just give the program the vocalization itself, which means you don't need to make assumptions about what about that vocalization is important. It's unsupervised, so you don't need to have labeled data. You don't have to tell it, I think there are this many vocalization types. And ultimately, these UMAP representations improve the clusterability of your vocalization. So what do I mean by that? What I haven't said up to this point is that the whole goal of creating these representations of vocalizations is that we want to then classify the vocalizations into different types or categories so that we can understand things like vocalization diversity. And so I just want to quickly illustrate the differences between traditional methods and methods like UMAP. So here, this is a principal components representation of some isolation calls from 12 different Egyptian fruit bats. Each color is a different individual, and each point is one call. The spectrograms bordering the plots are just showing you what representative calls look like from different regions. And you can imagine that if you were to throw some sort of clustering algorithm at this, it wouldn't do very well. You have all of these points forming kind of an amorphous blob, and it's hard to tease things apart. That exact same data, when you actually use UMAP to represent it, so you give it the spectrograms, let UMAP do its thing, we start to see some individual specific clusters emerging here. And once again, we didn't have to tell the program what features to pay attention to. It found the features it thought was most important, and that allowed it to separate the data. So all of that to say that for species like bowhead whales and bottlenose dolphins, where there is spectral and temporal complexity, UMAP is a really nice way of dealing with their vocalizations. And that's some of the work I've been doing here at OSU. So again, we want to make these representations of vocalizations so that we can then quantify diversity of vocalizations. And now I'm going to tell you about what we've learned for those 3 different species by doing this, starting with sperm whales. Before I really dig into it, I just want to give a little background on sperm whale social structure. So the first thing you need to know is that it's a hierarchical social structure. At the base of that hierarchy are individual whales. These individuals live in matrilineal social units that are made up of adult females and juveniles. The male sperm whales will disperse in their teens, but females stay in their social unit for life. And calves learn all of their behavioral repertoire from the whales in their social unit. Different social units will come together to associate for hours, days, or weeks at a time, and this forms what we call a group. But importantly, those groups are limited to whales that communicate using the same dialect of codas, so they use the same coda types. And those preferential interactions based on dialect segment the sperm whale populations into what we call vocal clans. Whales from different clans will never interact with each other, even when they live in the same water. And we believe that these vocal clans are distinct cultures. But to understand what we mean by that, of course, we have to talk about how we define culture. I am an academic child of Hal, so I define it how he does, which is culture is information or behavior that's shared within a community and acquired from group members through social learning. And what we found with sperm whale studies, particularly off the Galapagos Islands, is that different clans not only use different coda dialects, but they also move and forage and socialize differently. So for this species, it seems like culture is a really pervasive and important aspect of their life. So with that background, again, the signal of interest here are codas. Complexity typically manifests in the temporal domain, and we represent their structure as interclick intervals. And we can then look at the diversity of possibilities by then applying some sort of clustering to that interclick interval representation. This is what I spent a lot of my PhD doing. Most of it really was just to be able to sort of say this last point. And so what I was working on with some collaborators was developing a new method for detecting structure in acoustic data sets, specifically using sort of a theory about identity calls, which we'll come back to. But the way this method works is that you first have to divide the calls in your data set. It doesn't have to be sperm whales into repertoires. So you could do that by field site where they recorded, by individual who they were recorded from, by the day, doesn't really matter. You then classify calls into types using contaminated mixture models. This is the part that took about 3.5 years to figure out. It works really well. I'm not going to go into the specifics of it, but I'm always happy to talk about this if you're interested. After calls are classified into types, we then hierarchically cluster repertoires of calls based on similarities in call type usage, which gives us a dendrogram or a tree of repertoires. And then we start to look for identity clades. So these are clusters of branches in the tree that use specific call types, and those call types are not used by other portions of the tree. And so the thought being that these identity calls and identity clades might tell us something about underlying structure in the populations that we're studying. What does that look like? The output from the ID call method is a 3-part figure. The first part is a dendrogram. So each branch in this case is a different day of coda recordings, in this case from Caribbean and Mediterranean sperm whales. Branches that are closer together use more similar types of codas. The output also includes a heat map where each row is a different coda type, and the gray shading indicates how much that type is used by each repertoire. The last thing the program does is it adds color to show any potential detected biological structure in the system. And so in this case, the program found at least putative evidence that there might be 3 sperm whale clans in this data set. Repertoires that are pretty uncertain won't be assigned to any identity clade, which is good. And the last piece of the puzzle is that we get this output showing the clan identity codas. So what you're looking at on the right there are sort of a dot plot representation of codas. Each dot is a click, and it's showing you what the rhythmic pattern of the coda is, colored by the clan that uses that call the most. An important thing to say is that there are other coda types, right? Those are the identity codas that we use to tell clans apart, but there are plenty of coda types not restricted to single clans called non-identity codas, which are shown in this plot. So you can kind of already see where the identity codas are. They're the really dark cells that are showing you how the program found the different groups. So we did this on this data set, found evidence of 2 clans in the Eastern Caribbean recordings, 1 clan in the Mediterranean, matched what we expected, great. Something important about this method is that though it was designed with sperm whales in mind, the underlying theory can be applied to other species. And we showed this by successfully using this method to segment wren songs into 2 different subspecies and cricket songs into 2 different species. So this isn't restricted to just sperm whales. But if we go back to this and we focus specifically on those 2 clans we found evidence of in the Caribbean, it's important to point out that the recordings we used here primarily came from 1 island in the Lesser Antilles. So that's the island of Dominica, indicated with the white arrow there. But in 2019 and 2020, Dr. Felicia Vachon, who was also a PhD student of Howell's, led sperm whale surveys throughout the Lesser Antilles. So the map on the right is showing 3 different vessel survey tracks, inshore, offshore, and a windward side. And she recorded, as you might imagine, many, many more codas from many different places. She took those codas and she plugged them into ID call and confirmed the presence of 2 Caribbean clans. Their identity codas matched the ones we detected in the previous study. But excitingly, she also found compelling evidence that there might be a 3rd clan in the region. So something we didn't know before. It became clear pretty quickly why. Because that clan, which we call EC3, is rarely ever seen off Dominica. So Dominica is that really bright blue. What you're looking at here is a kernel density plot showing the Eastern Caribbean clan locations based on acoustic encounters. In gray are the vessel tracks where our boat was. And then we can also look at this in a slightly different way. So this plot is showing the movements of photo-identified sperm whales in different years. The dots are when we photo ID'd that animal. Solid lines are movements across the 2 years. Dashed lines are movements within. And what you can see is that the Eastern Caribbean 1 clan, the whales in blue, are basically bypassing Martinique and St. Lucia. So it seems like there's some cultural habitat partitioning that's happening among these 3 clans. So these analyses have helped us to better understand sperm whale cultural diversity in the Caribbean Sea. The area spanned about 700 kilometers. But we wanted to know if they could also help us understand sperm whale cultural diversity and clan movements on a much larger scale, in this case, throughout the Pacific Ocean basin. So using codas recorded from 23 different Pacific locations, we set out to answer this. As you might imagine, this was a group effort, so it was made possible by data contributions from a consortium of Pacific sperm whale researchers. And again, we fed this large data set into the ID call program. Just to reiterate, this is the dendrogram that we got. Each branch is a repertoire, in this case, all the codas recorded on one day in one region. Closer branches, more similar repertoires. Here we have a heat map, again, where each row is a coda type. The darker the cell, the more that coda type is used in that repertoire. And in this case, the program detected 7 clans in the Pacific. So before this study, using smaller data sets, we thought there were 5 clans. Now we have evidence for 7. And we can once again get the identity codas that are made by each of those clans, which we named based on kind of the predominant rhythmic pattern in their identity coda types. We also have this measure of repertoire to clan confidence. For the geneticists in the audience, if this reminds you of structure plots, that was the direct inspiration for this. So the idea being that for each repertoire, we kind of wanted to assess how likely was it to be from the clan we thought it was versus from others. As you can see, the clans on the left and right sides of this dendrogram look a lot cleaner than the clans in the middle. So this tells us where we have less certainty. And this work is in PLOS One, if anyone's curious about it, but it is heavily based on the structure program for genetics. And if you're thinking, wow, those figures look way prettier than any of the other ones you've shown us, that's because Mauricio made them. So just a thank you for that. Okay, so we have 7 detected clans in the Pacific Ocean. That means that we can start to look at the distribution of these different whales. Here is a map. Each point is one repertoire. The colors, of course, correspond to the different clans, and the size of the point indicates how confident we are that we got the clan assignment right. So you can see for the bigger points, we feel pretty good. For something like off of Palau, we have very little confidence that we actually know who these whales are. Often this happened because we didn't have many codas recorded in those regions. That being said, we can still draw some sort of big conclusions from this work, including on clan spatial scale. For example, the +1 clan was only ever encountered off of the Galapagos Islands in mainland Ecuador. That's about a 1,000-kilometer range. This is an order of magnitude difference than a different clan, the short clan, which spans the entire Pacific Ocean basin. And so now we can start to ask cool questions about why that difference exists. But we can also draw some inspiration from what we've seen in humans too. So we now know, based on this work, at least in the Pacific, that sympatric clans are the norm, not the exception. So that means that most regions had at least 2 different sperm whale clans. And if we look at something like this map, which is showing ethno-linguistic groups of Southeast Asia, we can see a lot of similarities. So not only are there multiple cultural groups in regions for sperm whales and for humans, some occupy really small regions, some occupy much broader ones, certain ones overlap more or less often than others. And so I think this is just kind of the tip of the iceberg in helping us understand how complex and far-reaching sperm whale culture is. And it points out some interesting similarities between our cultures and theirs. Now, if we go back to those identity codas, as a reminder, these are coda types that are used almost exclusively by specific clans. And we can exploit those differences in use to tell different clans apart in recordings. But the next logical question is, okay, are the sperm whales using these coda types in the same way? And if you talk about something like that, you inevitably fall onto the topic of symbolic markers. These are arbitrary traits that broadcast cultural group membership and modulate different individuals' interactions. They're extremely well documented in humans and can include things like accent or dialect, which is obviously relevant for sperm whales too. And they're generally considered a hallmark of human culture, so part of what makes us humans. But it's much less clear if other animals use them as well. And just to kind of illustrate the power of something like this, I grew up here in Pennsylvania, not here, US here, but I grew up in Pennsylvania. If you ask me what I call this article of clothing, I would say a hoodie. But I did my PhD in Canada, and if you asked almost any Canadian what they call it, they would also say a hoodie, except if the person is from Saskatchewan. in which case they call it a bunny hug. And so this is a really crazy thing, because from hearing one person say one word, bunny hug, you can already start to get a guess as to some cultural groups that that person considers themselves part of. So they probably consider themselves Canadian. They consider themselves Saskatchewanese. I don't know what it is, but that's. Right, so symbolic markers, very powerful. And they give you a lot of information from not a lot of words in the case of humans. We do think that identity codas are evidence of symbolic markers in humans. And I'm going to explain why by showing you two plots. So for each pair of clans in the Pacific Ocean, we calculated how much their ranges overlapped, which is the x-axis, and how similar their coda dialects were, which is the y-axis. But we did that separately for identity codas and for non-identity codas. And to remind you, those non-identity codas are ones that the program couldn't use to tell clans apart because they're used by multiple clans. If symbolic markers are happening in these identity codas, we would expect that as clans become more overlapped, their acoustic similarity goes down. This is based on really well-established research in humans. And the reason is because if you have multiple cultural groups heavily overlapped in an area, you get this enhancement of the things that make you different so that you won't confuse the cultural groups. In contrast, we wouldn't expect to see any trends in the non-identity codas. So you're about to see a bunch of these beach balls on the plot, and it's just showing you the two colors are the two clans that are being compared. And so, for example, in this case, the two clans are 4+ and rapid increasing. Their clan spatial overlap value is about 0.5, and you can see their acoustic similarity. So we do that for all of the clans. Sure enough, we do have this trend where we see that identity coda similarity decreases as clans become more overlapped and doesn't significantly change for non-identity codas. So we think this is evidence of symbolic marking in sperm whales through the use of identity codas, at least in the Pacific Ocean. But this is a globally distributed species, so we want to know if that extends. And so we actually have access to sperm whale recordings from 50 different sites across the Atlantic, Pacific, and Indian Oceans. This is an unprecedented data set of over 72,000 codas, and we're currently analyzing it using some of the methods that I talked about earlier. As many of you know, unprecedented data sets can only come from unprecedented collaborations, and I feel very lucky to be part of this one. And so because this is ongoing, I'm just going to do the results teaser, which is that it seems like culture does indeed structure sperm whale populations on a global scale. And for those of you who will be in Perth at the upcoming Marine Mammal Conference, this is work that I'm going to be presenting there. Why does any of this matter? Great question. Culture, we are increasingly recognizing, has significant implications for how we conserve species for which it's important. And a lot of that comes down to the fact that for cultural species, social learning is really important, and different individuals in a population can hold more or less knowledge than others. So we're starting to see that animal culture is really important for conservation. Sperm whales are one of basically 3 sort of keystone species that are being used to argue this. And so this analysis at these various different spatial scales of seas and ocean basins and globally are just helping us show why this work is important and that animal cultures are more widespread than we previously thought. But we're going to migrate now to much colder waters. Bowhead whales are the next ones up. They're an animal of extremes. It's the only baleen whale that is historically endemic to the Arctic. They can live over 200 years, and they spend much of their life under or at the edge of the ice. And so this species really perfectly exemplifies the merits of using acoustics for monitoring, because they're obviously very remote and difficult to access, but they also vocalize year-round. So for this species, the social vocalization I'm talking about is song. Again, it's a hierarchically structured vocal display. So you have single notes or units that are repeated together in phrases. Those phrases are strung together to make up a song. Songs themselves are repeated in song bouts, which in turn are repeated in song sessions that can last many hours. Hopefully it's clear from looking at that we have complexity in both the spectral and temporal domains in this signal. And that means that UMAP can be a really great tool for trying to represent the different parts of bowhead song. Specifically, my work is starting at the base of that hierarchy and trying to see if we can use UMAP to represent song units. And then we can use some sort of clustering algorithm on the UMAP representation to look at song unit diversity. To give you a little population information, there are currently 4 recognized stocks of bowhead whales, all of which were hunted to commercial extinction in the 20th century. I'm going to focus on 2 today. So the first is the Bering Chukchi Beaufort stock. There's about 17,000 animals now, so these guys have recovered pretty well from commercial whaling. And they're an important cultural, spiritual, and nutritional species for Inuit communities in the region. The Spitsbergen's bowhead whales, that stock is about 2 orders of magnitude smaller. So we think there's maybe 300 animals, but it's hard to get a proper estimate on their population size because they're extremely ice affiliated. So they don't get pushed south in the winter as the ice is growing, but can spend parts of that time hundreds of kilometers under, like inland under the ice. Inland, like the ice is here and they're 100 kilometers under the ice that way. And so Kate and colleagues published an analysis of Spitsbergen's bowhead whale songs in 2016. They used a hydrophone mooring in Fram Strait to record bowhead song and found something really remarkable. So over just a 3-year period, this extremely small population of whales produced 184 distinct song types, which is just to say, again, we have maybe 300 animals producing this crazy diversity. Most songs lasted only a few hours to a few days. And the speed of song turnover, coupled with the diversity of song types, is pretty remarkable for a mammalian singer. So that, of course, begs the question, do other bowhead populations show similar levels of song type diversity and evolution? Specifically, we're looking at BCB bowhead whales. So from late October to early May, these animals move through the Bering Strait for their fall and spring migrations, and they sing. So we have a hydrophone that is kind of located right in that migration pathway, which has given us a really awesome data set spanning 11 years. We're specifically looking at 4 migratory seasons during the peak months of bowhead singing, so during December and January. We also have some variation in sea ice conditions during these migratory seasons, which could add some interesting nuance to our results eventually. And we have sort of 2 tiers of analyses planned. The first is a song level analysis where we manually classify songs into types. Now, this involves looking at and listening to lots of hydrophone recordings of songs, annotating as many individual songs as possible, and then deciding ourselves if we think they're the same song type or a different song type. I spent the whole first part of the talk talking about how great automated methods are, but bowhead whales are also the most complicated, tricky species I've ever studied, and they really don't make it easy. So we have a lot of help through undergraduate researchers. Nikita is a student who is here this summer, who's continuing to help with this work. But never fear, we are going to try to do some automatic stuff. We're specifically, like I mentioned, also looking within songs at the units to try and assign units to types automatically using UMAP. And the goal of that, oh, Nikita's helping with that. And the ultimate goal for bowheads will be something like this. So here you are looking at Bengalese finch song sequences. These are song syllables. Each dot is a syllable from a single animal, colored by syllable type. So we made a UMAP representation, clustered it, and got these syllable types. So imagine we're doing this for bowhead whale song units instead. The next thing you can do is you can look at transitions between different categories of units and represent those transition probabilities as a Markov model. You can then take songs themselves and basically conceptualize them as a string of these different categories. And if you do that for many, many different songs, you can start to visualize changes in the pattern structure of songs. So this is what we eventually hope to do with bowheads. But in the meantime, we're using the tried and true methods coupled with some of this novel work. Why is this worthwhile? So first off, this would be a major methodological advancement if we're able to get UMAP working well on this species. They have notoriously complex and tricky songs, and they're singing them in a very noisy place. This is also a first step towards determining whether population level song differences exist, for example, in Spitsbergen's bowhead whales and BCB bowhead whales. If there are differences, we can exploit those differences to track movements of individuals from specific populations, especially in a changing Arctic. So the 4 populations used to be isolated from each other because of sea ice, but with climate change-induced declines in sea ice, we are seeing that whales from different populations can be in the same place at the same time. Finally, going back to the title of the talk, this type of work will also help us understand what information is being conveyed in these songs. So one question is this crazy diversity something that's unique to the Spitsbergen's bowhead whales, or something more indicative of what it means to be a bowhead whale generally? And if it's the latter, we want to know, is song in bowheads a cultural trait, meaning that it's socially learned and transmitted? And so we think the answer is probably yes. You don't get these really short song lifespans typically without some sort of social learning happening. But to answer that, you need long time series of data. Finally, this can help us better understand the evolutionary pressures that are being applied to a display like song. So is there some sexual selection? Is there pressure from females for songs from males, for males who can innovate or change more rapidly? Things like that. And those all sound like pretty ambitious goals, but I do want to point out that this work has, for the most part, been done for another species, for humpback whales. Male humpback whales sing complex songs in mating contexts. If you want to learn more about the mating context, then you should be here in 2 weeks for Franca's talk. She's going to cover that as well in a seminar. But one of Franca's PhD supervisors, Ellen Garland, has tracked the spatial and temporal dynamics of humpback song in the South Pacific. So this plot is showing humpback song in 1 year, 1998. Different colors indicate different song types. Hashed cells don't have any data. And we can see in this 1 year that the whales in Eastern Australia, New Caledonia, and Tonga were all singing the black song type, whereas further to the east, the whales were singing this pink song type. We can do that for another year. We see that in 1999, all of the whales in regions that they had data were singing the same song type. And if we do this over an 11-year period, we can see that multiple song types spread rapidly throughout the population, rapidly being over the spans of 1 or a few years, and that it always went from west to east. And since 11 years is much shorter than the lifespan of a humpback whale, it seems like there must be some social learning happening. And in the decade and more since this work was published, we know that this is a cultural display, that it's horizontally transmitted from males to males in the population, and that part of the reason we see the change over time is because there is an evolutionary pressure for novelty. So this type of work can be done. Humpback whales live in really warm waters that people like to go to, and bowhead whales are a bit harder to access. So I think it's an ambitious goal, but we have a lot of long-term recordings now, so we can start to do this for that species too. But moving to warmer waters, for the last species I'm going to talk about today, that is the bottlenose dolphin. These animals produce whistles in social situations. Complexity is in the spectral and the temporal domain, which again means we can use UMAP to create a representation of these whistles, and we can then cluster that representation to figure out whistle diversity. The system that we're interested in is one that many of you are familiar with. This is a cooperative fishery between human and bottlenose dolphins in Laguna, Brazil, where artisanal fishers and dolphins work together to catch mullet. And the way this works is that when the mullet are migrating in certain seasons, they move into this narrow channel. The dolphins will herd the mullet towards a line of fishers, and then the dolphins perform a very specific body or head movement, which the fishers take as a cue to cast their nets. And if this interaction goes well and both parties are sort of on the mark, it means that everyone catches more fish than they would if they were working alone. Now, this cooperative interaction has been the subject of a long-term study. Mauricio is one of the PIs on it and has led it, and it's extremely well characterized. So every year, researchers go to the field to collect data on things like sonar footage of the mullets, photographs of the dolphins themselves so we can identify them, the catch rates of the fishers, and so on. Of interest to me, especially are acoustic recordings of the dolphins that are doing this. In the past, the team has primarily used a single underwater microphone, a single hydrophone, to record these animals. But in the best case scenario, what we'd actually like to do is use an array of hydrophones, because doing that, using multiple hydrophones, means we can localize the sounds in the direction of animals producing them. That's what we attempted to do for the first time this past field season. So in May and June, I was part of the Laguna team that tested out a small portable array setup. And so you can actually see this is a line of fissures, and then our array, like a little fissure in the lineup. And so at the base of it, we have 4 hydrophones that are arranged in a tetrahedron. And what happens is that when a dolphin whistles, that whistle will be detected on all 4 hydrophones, but one of them will be closer to the dolphin. You know, the sound is not actually going to reach each hydrophone at the exact same time. And you can use that difference in sound arrival to calculate an angle, which can give you a guess as to which animal made the sound. Those hydrophones are there, and they're connected to a multi-track recorder. And we also listened in real time and took notes on the dolphins we could see, so that we can hopefully correlate the measurements that we get from the array with what we saw in the field. This is in collaboration with several of Mauricio's longtime colleagues and their students. So Fabio and Pedro designed the array. Bianca did her PhD on communication in the system, supervised by Fabio. And Hannah is a new graduate student who's actually working on creating a catalog of signature whistles for dolphins in the region. Signature whistles are specific whistle types that bottlenose dolphins learn early in life and use throughout their life as an individual identity signal. The closest comparison we can draw is to human names. In this picture, you can see the signature whistles of 28 dolphins in Namibia. And the main takeaway is that these whistles are different. They differ for each animal. And dolphins often produce these when they're being reunited with other individuals. But bottlenose dolphins also produce a ton of other whistles, which we collectively just call non-signature whistles. Those are actually 70% of the whistles that a dolphin produces. So signature whistles and then everything else. And we don't know so much about that everything else. But one thing that is sort of a long-term goal of the project is to investigate whether through whistles, we can figure out whether the dolphins themselves are cooperating with each other when they're doing these fishery interactions. This is a key unresolved question. Your first thought might be, oh, you said you have drones. Why can't you just, take the drone up, look down? And obviously, for these animals, a lot of the action is happening under the surface. So our thought is that we can quantify whistle production and whistle diversity in different situations. For example, when 2 dolphins are in the bay, but only one is cooperating with the fishers, versus 2 dolphins in the bay, but both cooperating with the fishers. And we can start to use that as a first proxy for understanding if there's cooperation within the dolphins and not just between dolphins and humans. So to wrap things up, if we go back to this goal that I said has kind of guided my career, I want to quantify spatial and temporal variation in social vocalizations, because doing so can tell us about the identities, interactions, and cultures of the animals that produce them. And so today we went through some of those examples for different species. So studying variation in codas has taught us about sperm whale population structure, cultural identity, and cultural movements. Studying this variation in songs is currently teaching us about bowhead whale population connectivity, bowhead whale vocal culture, and the evolutionary dynamics on bowhead song. And studying this variation in whistles in the future will teach us about dolphin individual identity, individual movements as well, and potentially their cooperative intent when foraging. And as I do this research, I continue to appreciate and often be humbled by the fact that the characterization and abstraction of vocal communication signals remains both an art and a science, and I hope you are too. With that, I want to say a huge thank you to the Labyrinto and Mammal groups who have put up with me being especially scatterbrained this week as I prepared for this. I am in the Marine Mammal Institute ecosystem, which is just about the best place to work, and I'm funded by the Oregon Gray Whale License Plate Fund. Thanks as well to Clarissa Teixeira, who helped me beautify this presentation, and to all of the organizations that have funded or provided in-kind support for some of the work I talked about today. Thank you as well for your attention, and I'm happy to take any questions.
Wow, that was impressive. So many person hours and person years involved in that data. I guess we will take any online questions or any coming in. All right. In that case, anyone in the room have a question? You can raise your hand and I'll pass you the mic. Right.
Hi, Taylor. Great presentation. I was wondering if you could explain a little bit more about the signature whistles, because one of the things that I always wonder about this is like, you said that the most similar thing would be our names. So they are basically repeating their names to other individuals, or that would be a way of like, I don't know. Can you explain?
Yeah, that's a great question. The closest approximation are names, but the use is a bit different because bottlenose dolphins will often produce their own signature whistle a lot, right? So they're just, it's like Clarissa, 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 and it's like, what's the point? They do sometimes make other animals' signature whistles, but we think that they are primarily important when they're being reunited. So a lot of the sort of best work for that has come from captive bottlenose dolphin studies where they have animals that have been separated for whatever reason, and then they're finally put back in the same pool, and you get a ton of these signature whistles. So it seems like it's an identity cue, and it's stable throughout their lives. It develops very young, and they maintain it. But in terms of, I mean, I guess what's the point of a human name? We tell people, that's maybe existential. But yeah, I think what your question is getting at is this really cool part, which is there are so many other whistles they make which aren't signature whistles, and what are all of those for? And so that's a question that I still have too.
All right. It looks like we have some online.
How much link is there between group bases based on calls and genetics? And do group members tend to be closely related to each other?
Can you say it one more time, please?
How much link is there between groups based on calls and genetics? And do you see members to be closely related?
I think I can answer this for sperm whales specifically, so I'll do that. So if we think about sperm whale social structure, the animals in a social unit are typically genetically related through the matrilineal line. So you have mothers and grandmothers and aunts and daughters. Groups that come together for sperm whales don't have to be closely related. And certainly we know from genetics works done at the clan level that because male sperm whales will mate with females from different clans, that kind of scrambles the nuclear genetic gene pool. There are some trends in mitochondrial DNA, which is from mothers. But yeah, for sperm whales at least, the genetic relatedness is happening at kind of like the more basal level of their population structure. Yeah.
All right. Oh, I see more hands popping up here. I'm not sure who was first. I saw your hand and I saw your hand, but.
Yeah, Taylor, lovely talk, really interesting, and lots of really cool methods you've applied on complex data. So good job. But as an ecologist, I think about these sort of things. I want to take it back to the ecology and behavior of these animals. So lots of your results show culture, and I'm a firm believer that all these populations have unappreciated culture within them. But when I think about it, I think about behavior or morphology or space use. And you did talk about the differences in space use relative to these different call or acoustic types or groupings. And particularly I was thinking about your example of the sperm whale where you have one large group across the whole Pacific and then one specific to the Galapagos region. And I just had, I guess what my question is for all of this, have you tried to, you and your colleagues tried to tie all these cool findings back to either morphological differences or behavioral differences or foraging niche differences that sort of can explain those different groupings and types?
Yes, so that's an excellent question. And for sperm whales, some of the bigger conclusions that we feel comfortable making are grounded in the fact that those multifaceted studies have been done in specific regions. So the Galapagos Island sperm whales are sort of the best example of this, for the Pacific at least, because there we know that the clans vocalize differently, but they also move differently. They have different foraging success quantified from poop rates at the surface. We don't actually know if they're targeting different prey, which is partly a problem of big squid beaks sink faster than small squid beaks. But yeah, the work in the Galapagos, which has been done at this very fine clan level, has shown that they also, the movement differently is like some clans travel in more straight trajectories and others wiggle more, which is probably relating somewhat to foraging. And so we're kind of using that as we haven't done that level of work on every clan that we think exists in the world, but for all the regions, including in the Mediterranean Sea and in the Caribbean Sea, where the clans are studied at a much closer level, we do see that behavioral variation across many different levels. And so that's why it seems like culture is important and it's affecting multiple aspects of their lives. But kind of the call to action, for example, at the end of this global paper will be, let's make sure we're right. This is a conservative estimate of the cultural diversity worldwide, and now we need to go and do those studies on these populations and see if we get these other variations in behavior as well.
All right. Katie, do we have any new? Okay, I'll head up here. Like Donahue.
Great talk, Taylor. Can you flip to that sperm whale, the short clan and the double whatever clan? My question's really similar to Lee's, but I'm going to ask it anyway. And it's, well, I'll wait till you pull that up.
Yeah. Oh.
Yeah, and then I think it's the next one.
Oh, right. Sorry, the difference.
Yeah. So what does it mean to have a clan that spans the Pacific? Like, what is that actually versus a clan that only exists in Ecuador? Like, what do you think? How do you think about what that means? Are they like small demographic groups that have some link where they, maintain the same sort of vocal structure and culture and things, and there's connectivity there? Or I just am not sure, especially after you said that there's not necessarily a genetic link or genetic relationship there. How do you interpret that? What does it mean?
Yes, great question. So many things to say. I will try and do it briefly. The first thing to say is that in the short clan, we don't believe that the short clan, Eastern Pacific short clan whales know the Western Pacific short clan whales. These animals aren't migrating in that way. They're not really migrating much at all, although at least in the Eastern Tropical Pacific, we know that the home range is like sort of 1,000 kilometers. So what I think has happened to get these kinds of trends where you have some clans that sort of span an ocean basin is that you're having kind of an older pattern of like cultural leapfrogging sort of. So some whales, I mean, we think the other fun thing is that we think the short clan dialect is basal. It's the rhythmically simplest codas that are produced. It's the most widespread clan off of New Zealand, where we just have male sperm whales. The codas they produce put them in the short clan. And so I think that something about the short clan is kind of a basal sperm whale state almost. And so when we say that a clan spans the ocean, it's what the data is showing us. Acoustically similar whales are in these different parts of the world. When you think about what it means for actually conservation and management, not much, because what we care about is location-specific places where, for example, in the Eastern Caribbean, where we know that one of the clans is bypassing St. Lucia and Martinique, and two other clans are almost exclusively using those. That can help us understand if we need to mitigate our impacts off of those islands. Maybe St. Lucia and Martinique are, they don't have currently sperm whale watching tourism like Dominica does. We can also, once we know that multiple clans are in an area, we can map population trajectories for different clans and see if they're doing equally well or not, which can lead to clan-specific conservation action. So I think that's what I would say.
Hi, Taylor. Thanks so much for your talk. I'm sorry if this has already been covered, and it possibly relates to my experience as an English person recently joining the US, but I wondered whether we see any ability of these sperm whales to learn, or whether they have any change in their identity calls across their lives. So I guess I was wondering whether there's any kind of spread. Maybe the more widely traveled whales might have less distinctive calls, essentially.
So the coolest thing I can tell you is that sperm whale calves babble codas. So sperm whale calves produce a greater diversity of coda types than the clan that they're born into will. So there's some sort of maturation within individual sperm whales that happens where by about 3-ish years of age, they're sort of speaking the dialect of their clan. But when they're younger, they're producing coda types that the clan does not produce. So that's a good reason to think there's some of the social learning happening. In terms of like whether whales that travel further have less distinctive dialects or not, I don't know that we know that yet. Again, I would think the whales from the Pacific analysis that we know are traveling far, that we have codas recorded from, are the New Zealand male sperm whales, because they're going to polar waters and coming back, so migrating more than females are. And there we do see this kind of pretty basic dialect. The short clan dialect gets its name because whales make codas with 3 or 4 clicks, and they're isochronous. So it's click, 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 or click, 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 click. So it's a pretty basic, acoustically simple signal. But yeah, I don't think we know beyond that.
Thank you.
Okay, it's 4:30, so I think we can start wrapping up, but I think everybody can stick around. There's more cookies and coffee and tea. And if you have more questions, I'm sure Taylor might be ready to answer them. But yeah, let's give her another round of applause for all that amazing.